Hi everyone, Gina Logrande here. Thank you for taking the time to view my presentation on aerobic respiration. I hope you enjoy it. What is aerobic respiration? Aerobic respiration is the aerobic catabolism of nutrients to carbon dioxide, water, and energy, and involves an electron transport system in which molecular oxygen is the final electron acceptor. Most eukaryotes and prokaryotes use aerobic respiration to obtain energy from glucose. The overall reaction is C6H12O6 plus 6O2 yields 6CO2 plus 6H2O. If you remember your chemistry, you'll recall that this is a combustion reaction. The process of aerobic respiration occurs in 4 distinct steps: glycolysis, which occurs in all organisms, prokaryotic and eukaryotic; pyruvate decarboxylation, known as the Bridge reaction, which sets the stage for aerobic respiration; the Krebs or citric acid cycle; and oxidative phosphorylation, also known as the electron transport chain, an oxygen-dependent proton gradient pathway that occurs in sequence in the mitochondria. The entire purpose of cellular respiration is to generate enough ATP to carry out the major metabolic processes of life. ATP is a nucleotide that consists of 3 main structures: the nitrogenous base, the sugar or ribose, and a chain of 3 phosphate groups bound to ribose. We will discuss nucleotides in more detail in upcoming slides. The phosphate tail of ATP is the actual power source which the cell taps. Available energy is contained in the bonds between the phosphates and is released when they are broken, which occurs through the addition of water molecule in a process called hydrolysis. Usually only the outer phosphate is removed from ATP to yield energy. When this occurs, ATP is converted to adenosine diphosphate or ADP, the form of the nucleotide having only 2 phosphates. As we learned in an earlier presentation, the first stage of cellular respiration is glycolysis, a set of 10 reactions that do not require oxygen and hence occur in every cell alive. Glycolysis takes place in the cytoplasm. During the investment phase of the process, 2 ATP are consumed as 2 phosphates are added to the glucose derivative before it is split into 2 3-carbon compounds. These are transformed into 2 molecules of pyruvate, 2 NADH, and 4 ATP for a net gain of 2 ATP. The second stage of cellular respiration, the transition or bridge reaction, gets less attention than the rest of cellular respiration. As its name implies, however, there would be no way to get from glycolysis to the aerobic reactions or beyond without it. In this reaction, which occurs in the mitochondria, the 2 pyruvate molecules from glycolysis are converted into 2 molecules of acetyl-CoA, with 2 molecules of CO2 produced as metabolic waste. No ATP is produced. As this reaction was also covered in an earlier presentation, we'll move on from here to the citric acid cycle. The citric acid cycle does not generate much energy in the form of ATP, but by combining the 2-carbon molecule acetyl-CoA with the 4-carbon molecule oxaloacetate and cycling the resulting product through a series of transitions that trim the molecule back to oxaloacetate, it generates 8 NADH and 2 FADH2, another electron carrier. The cycle occurs in the mitochondrial matrix. These molecules are needed for the electron transport chain, and in the course of their synthesis, more CO2 molecules are shed from the cell as waste. In the final stage of aerobic respiration, electron transport is used to power the transport of protons, leading to the production of ATP. This process is known as oxidative phosphorylation. The protein complexes at the start of the chain accept electrons from NAD and reduced FAD. These electrons are then passed along the chain from complex to complex. As they move along the chain, the electrons attract protons, drawing them out of the mitochondrial matrix through the inner membrane and into the intermembrane space. The final protein complex, cytochrome oxidase, passes the electrons to oxygen, forming water, together with protons present in the mitochondrial matrix. Because the protons drawn into the intermembrane space are unable to penetrate the membrane and return to the matrix, the concentration gradient across the membrane increases dramatically. This gradient powers ATP synthase. The protons rapidly return to the matrix through ATP synthase, rotating a pair of motors that produce ATP from ADP, an inorganic phosphate. This brings us to the pentose phosphate pathway, an alternative route for the metabolism of glucose. It doesn't lead to the formation of ATP, but has two major functions: the formation of NADPH for synthesis of fatty acids and steroids and maintaining reduced glutathione for antioxidant activity, as well as the synthesis of ribose for nucleotide and nucleic acid formation. It is a more complex pathway than glycolysis. 3 molecules of glucose 6-phosphate give rise to 3 molecules of CO2 and 3 5-carbon sugars. These are rearranged to regenerate 2 molecules of glucose 6-phosphate and 1 molecule of the glycolytic intermediate glyceraldehyde 3-phosphate. Since 2 molecules of glyceraldehyde 3-phosphate can regenerate glucose 6-phosphate, the pathway can account for the complete oxidation of glucose. The most important thing to understand about the PPP is this: the pentose phosphate pathway forms NADPH and ribose phosphate. Following the formation of PRPP from ribose 5-phosphate, the purine synthetic pathway is initiated. 9 enzymatic steps follow to produce the primary product, inosine 5-monophosphate, also known as IMP. IMP can then be converted to adenine and guanine nucleoside monophosphates, the purines commonly found in DNA and RNA. What are nucleotides and why are they important? A nucleotide is one of the structural components of DNA and RNA. A nucleotide consists of a nitrogenous base, which is composed of one of 4 molecules: adenine, thymine, guanine, and cytosine, plus a 5-carbon sugar called a pentose, and 1 phosphate group. Cytosine, thymine, and uracil, which replaces thymine in RNA, are known as pyrimidines, and each has a single nitrogen-containing ring. Adenine and guanine are known as purines, and each has 2 nitrogen-containing rings. How are they made? There are 2 ways nucleotides are synthesized in our cells. The first way is called de novo synthesis, which is the synthesis of nucleotides from simple molecules including carbon dioxide, glucose, ammonia, bicarbonate, and amino acids. The second way nucleotides are synthesized is through salvage pathways. Salvage pathways recycle nucleotides obtained from our diets or from degraded DNA and RNA to form new nucleotides that are needed by our cells. These synthesis reactions occur in the cytoplasm and are catalyzed by enzymes. Both deoxyribonucleic acid and ribonucleic acid are made up of nucleotides. They both contain a sugar and a phosphate, but have nitrogenous bases that are different sizes. Because of this, the 2 different groups are synthesized in different ways. However, all nucleotide synthesis requires the use of PRPP, which donates the ribose and phosphate necessary to create a nucleotide. This is the connection between the pentose phosphate pathway and nucleotide synthesis. As mentioned on an earlier slide, adenine and guanine are the 2 nucleotides classified as purines. In purine synthesis, PRPP is converted to inosine monophosphate or IMP. Production of IMP from PRPP requires glutamine, glycine, aspartate, and 6-ATP, among other things. IMP is then converted to AMP, adenosine monophosphate, using GTP, guanosine triphosphate, and aspartate, which is converted into fumarate. While IMP can be directly converted to AMP, synthesis of GMP, guanosine monophosphate, requires an intermediate step, in which NAD+ is used to form the intermediate xanthosine monophosphate or XMP. XMP is then converted into GMP by using the hydrolysis of 1 ATP and the conversion of glutamine to glutamate. AMP and GMP can then be converted into ATP and GTP respectively by kinases that add additional phosphates. ATP stimulates production of GDP, while GDP stimulates production of ATP. This cross-regulation keeps the relative amounts of ATP and GTP the same. Excess of either nucleotide could increase the likelihood of DNA mutations, where the wrong purine nucleotide is inserted. Pyrimidine nucleotides include cytosine, thymine, and uracil. The synthesis of any pyrimidine nucleotide begins with the formation of uridine. This reaction requires aspartate, glutamine, bicarbonate, and 2 ATP molecules to provide energy, as well as PRPP, which provides the ribose monophosphate. Equal amounts of purines and pyrimidines are required for DNA synthesis. In summary, aerobic respiration uses oxygen to break down glucose, amino acids, and fatty acids, and is the main way the body generates ATP, which supplies energy to fuel all metabolic processes in the body. After glycolysis, the anaerobic breakdown of glucose into pyruvate, pyruvate is converted to acetyl-CoA in the matrix of the energy-transferring mitochondria via the bridge reaction. Then the citric acid cycle, which occurs twice per glucose molecule, produces, among other chemicals that feed into the aerobic part of the process, more ATP. The aerobic part of the process depends on a series of protein complexes that are organized along the folds of the inner membrane called the cristae of the mitochondria. These are arranged so that the electrons pass from one reacting molecule to the next in a series of steps known as the electron transport chain. This process ends with ATP synthase, an enzyme that produces ATP from ADP and inorganic phosphate, up to around 30 molecules of ATP per molecule of glucose, according to current thinking, capturing the final portion of the energy released by the whole process in a form that the rest of the cell can use. This wraps up my final presentation on aerobic respiration. As always, I want to thank you for taking the time throughout this class to watch all my presentations. I know this one was long, and I appreciate your patience. I wish all of you the best in your next adventure, and I've enjoyed getting to know you all.